Ladies and gentlemen, please welcome to the Discovery Showcase stage Dr. Hao Chen, Professor of Chemistry and Environmental Science at the New Jersey Institute for Technology.
Good morning, everybody. First of all, I want to thank all of the Discovery Showcase organizers for this great opportunity to talk about our work that we recently working on at NJIT. So today I'm going to introduce you an interesting reactor which utilizes small water droplets for reaction. And thanks to the collaboration with Agilent Technology and Johnson, so we applied this reactor to quickly digest antibody, which is a very large molecules. And then so we can analyze the structure of antibodies. So the microdroplets reaction, was discovered by Professor Graham Cooks at Purdue University about 14 years ago. And so basically, most of the chemists doing reaction carry out reactions in a glass flask, but the reaction, could be slow. And however, if you do it in small droplets, where the droplet is small enough, like with diameter less than 10 micron, then the reaction can be greatly accelerated with acceleration factor up to a million times. So for the reason, probably in the confined volume like microdroplets, reactants can find each other much more easily, because in the microdroplets, the reactants, they tend to distributed on the surface. On the surface, they can easily find each other to collide with each other, so reaction can occur. However, in the flask, in the three-dimensional flask, it takes more time to find each other to react. That's why reaction in the microdroplets can be accelerated. And this phenomenon now has been gradually accepted by our chemistry community. There are a bunch of amazing reactions has been performed in microdroplets, and with benefits that the reactions can be done instantaneously, almost takes no time. For example, the reaction can be completed in microseconds instead of hours or days. So this slide shows some of the reactions that's very important to climate change, such as CO2 reduction, and energy problems such as nitrogen fixation to convert the nitrogen in air into fertilizer. And also people use this reaction, this faster reaction, to degrade environmental pollutants such as PFAS, toxic environmental pollutants. So we were thinking about the proteins. Can biomolecules be undergo reactions in microdroplets? So as antibody is very important, we know that this drug market is very big, as you can see numbers here in this slide. So many pharmaceutical companies, they develop new and novel antibody drugs to treat all kinds of disease. However, for quality control of those drug products, we need a method which can be quickly implemented to analyze the structural change or a possible degradation of those antibodies to secure the quality of drugs for the benefits of patients. So I got a chance to work with Stanford Professor Dick Zell, one of the top chemists in the world. And so when we mix a protein with enzyme and use a nebulizer to spread out and generate microdroplets, we find that enzyme can be instantaneously digested within within 1 milliseconds. So we also tried, during the COVID time, we tried the DNA digestion. It also can occur very quickly in microdroplets. As shown in this slide on the right side, we also tested converted RNA of COVID-19 into DNA. Then we can apply the nuclease to quickly digest the DNA within 1 millisecond. But and then we at the NGIT we thought about how about larger proteins like antibodies. So a few years ago, my collaborator, Dr. Harsha Guanawandina from Johnson, he urged us to test the larger proteins such as antibodies. So we tested this reaction. So simply mix like the antibody with enzyme and use a homemade nebulizer, and we generated microdroplets, and then we connected the sprayed droplets. And during this spray process, antibody got digested, and we know that by subsequent mass spec analysis, as shown in this slide, you see the mass spec data for the antibody before and after the spray. So the top panel shows the antibody fragments after digestion by enzyme. The bottom one is the intact antibody. So they are very different, as you can see the contrast. This experiment was done like using homemade device and done offline. So actually, even though the reaction takes place very fast, but everything is still was manually performed. So the whole process is still slow. And then Dr. Guanawandina, and he realized that agent technology has a very interesting, very good instrument, which is called a QTOF. And the QTOF is connected with LC, which is liquid chromatography instrument. And then he realized that if we introduce antibody and spray antibody together with the enzyme in the agent instrument using their ion source is called jet stream spray source, then the reaction can be completed. So, and also the sample, it can be automated. Sample injection can be automated because the agent LC has an autosampler which can do liquid handling. So by doing this, we can complete one sample analysis within 2 minutes. And later on, people further improve the analysis process. They can analyze one sample within like 10 seconds. So as you can see, like the engineering, the automation and also chemistry come together can really facilitate the protocol for drug molecule analysis. In this case, is antibody drugs. So this video shows how the agent instrument ion source works. As you can see, there is a nozzle on top, and you spray liquids, generate a very small mist. Those are microdroplets in micron size. So in this micron size, the droplets reaction can occur very rapidly. Although we don't know exact mechanism how enzyme can digest the enzyme so quickly, but we can see the products. We can see the disappearance of antibody peak, and as a result, we got the subunits of antibodies. So this and then agent was very generous and was willing to put a big instrument in our lab, so as a toy for us to play. So yeah, both me and my students are excited to further optimize this protocol. So we try to do different experiments, such as different enzymes, different antibodies, because in industry people really want to they don't want to use like a standard antibody, and they want to do engineering on antibody sequence to change the structure so the antibody can be purposed for different disease treatment. One of them is like they can do the FC, which means fragment crystallizable region. They can do the FC silencing, so that way it can enhance the half-life time of the antibody drug, but also can reduce side effect. so can avoid to protect patient. So those enzymes, mutated enzyme, actually cannot be digested using traditional enzyme like IDase. And then recently working with Argentine scientists, and we were able to find another new enzyme called fabricator extra. Extra enzyme can be able to digest the antibodies with different mutations, such as the mutation the residues to 34 to 35, they change from leucine into alanine in a very small, like a little change in structure, and the original IDase enzyme failed to digest this mutated antibody. However, extra enzyme can achieve very high digestion efficiency, up to 80%, as shown by this data. As you can see, the intact antibody peak is very small now, and also in contrast, the antibody fragment peak arose. And then we were able to test different antibodies, such as the drug like vidofludimus, and also a very famous anti-PD-L1 antibody. This antibody is known to be very difficult to digest, but we still can achieve very high, like over 80% of digestion efficiency using extra enzyme. So this diagram summarizes the results, like the traditional IDase field. However, extra can do the digestion, but if in the solution, it takes like 2.5 hours to get 80% of digestion efficiency. However, in the microdroplets, we only need like less than 1 millisecond. So digestion can occur in microseconds, and we can achieve equivalent digestion efficiency. So the reaction rate actually enhanced by 9 million-fold in this case. So right now, we also work with like another a company in Switzerland, and they provide smart enzyme. They do engineering for the enzymes so they can have different functions. So that way, so this is a summary. Basically, the engineering instrument with their special jet spray ion source can do the can affect the microdroplets digestion of antibodies, and extra enzyme can do the very efficient cleavage of antibodies. So now taking this opportunity, I also want to show you a couple of different projects in our lab at NJIT, so related to environmental analysis. So as you know, PFAS is a big problem in the environment. It pollutes our water, drinking water, soil, as well as our consumer products, such as all kinds of food packaging material, or even toy played by children. So to tackle this problem, one way is to do detection, fast detection. However, EPA method, the current EPA method takes a long time, it takes hours, because PFAS exists in trace amount. So people, in order to detect them, they need to do enrichment, and then followed by separation. It takes hours for one sample analysis. And then in our lab at the NJIT, we were able to detect one sample within 3 minutes, because we don't need to do a sample treatment. For example, we use a piece of triangle paper, apply high voltage and solvent. We can put a sample directly on the paper, such as drinking water or soil, or even the food packaging material. We can cut the food packaging material into triangle-shaped paper, and then we apply high voltage and solvent. We can instantaneously detect PFAS from those different samples with different matrices. We see this kind of help to accelerate the analysis process. Another work was about microplastics and nanoplastics. This is another class of environmental pollutants of great concern because they exist everywhere. Since we have used plastics for many, many years, and now is especially in the ocean, the fish got plastics, and we eat fish, and it's transferred to our body. So the biological effect of micro or nanoplastics is still unknown, but people know it can cause some problems, like or maybe Alzheimer's disease or like inflammation. And but the detection of these molecules this type of pollutant is not easy. So it could take days using the traditional current method. Because people need to digest the sample, like a biological sample, they need to run digestion, separation, and then so on. So it takes like several days for one sample, and it's very, because it involves so many steps, also the results is not very reproducible. And in our lab, we thought about this idea, so we just use flame. We can use flame to burn like a soil containing plastics or the tissue, biological tissue. We can just use flame. The flame temperature is high enough, it can crack the polymers, like plastic polymers, into small molecules. And also, flame has high enough temperature to ionize those resulting small molecules to become ions. And we do this in ambient conditions, just right in before the mass spectrometer, so we can do this in 10 seconds. And so we just recently published a paper and filed a patent pending to NJIT. So yeah, we thank the funding agencies, NICF and NIH, for the support. Also thank my students, Dr. Perry Zhao, Dr. Ronglin Ai, Rongqin Yang, Meng Yuan. They actually worked on the antibody digestion work with my collaborator, Dr. Gunnar Wandina from Johnson & Johnson. And I really thank my colleagues in engineering technology and also their instruments for us to use. And some of the engineering collaborators are in the audience. We really appreciate they spend their time to come here, even though they are very busy. And also, thank you very much for your attention.